Transcription is the process of synthesizing RNA from a DNA template. In the nucleus, the transcription pre-initiation complex assembles around the core promoter of a gene, which includes a general transcription factor binding site, here a TATA box, a binding site for the RNA polymerase, and the transcription start site. Once the necessary components are bound, the pre-initiation complex unwinds a short stretch of the DNA upstream of the transcription start site, and RNA polymerase begins producing a new strand of mRNA. Nucleotides are added one by one, and synthesis of the mRNA occurs in a 5' to 3' direction, reading from the template strand. This newly created strand of mRNA represents a copy of the information in the coding strand, except the thymidines are replaced by uracils. Synthesis will continue until a termination sequence is encountered, which will release the newly made mRNA and allow for further processing. Translation is the process of synthesizing a protein from a messenger RNA template. This process begins when the small subunit of a ribosome, coupled to a methionine tRNA, binds to the 5' end of an mRNA. The small subunit then starts scanning in a 5' to 3' direction until it encounters an AUG codon that will serve as the translation initiation site. Next, the anticodon of the methionine tRNA will pair with the AUG codon and recruit the large subunit of the ribosome. Synthesis of the new protein starts when a tRNA carrying its amino acid binds to the next codon of the mRNA through its anticodon. This action places the new amino acid in close proximity to the previously incorporated amino acid, which forms a peptide bond between the two amino acids. Translation continues as the ribosome moves to the next codon in the sequence. This forward movement is called translocation and will continue until the ribosome encounters a stop codon. Stop codons are unique in that they do not have tRNAs. Instead, proteins called release factors bind to the stop codon, which induces the ribosome to release the newly synthesized protein and causes the ribosomes to dissociate.